Hello, everyone, and welcome to today's webinar, Unveiling Tumor Immune Landscape Using Multiplexed Whole Tissue Imaging and AI-Powered Spatial Analysis. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. And with that, I'd like to now welcome our speaker. Joining us today, Vasundhara Agrawal, Applications Manager, Spatial Biology Image Analysis at Leica Microsystems. For a complete biography on our speaker, please click on the Presenter tab. Vasundhara, thank you for joining us. You may now begin your presentation.
Thank you for that introduction. Hello, everyone. Welcome to today's webinar. I am Vasundhara Agrawal, and I'm the Applications Manager for Spatial Biology Image Analysis at Leica Microsystems. And I'll be giving a talk about unveiling the tumor immune landscape using multiplexed imaging and AI-powered spatial analysis. And today I'll be sharing some key image analysis insights of tumor immune interactions from cancer models. So let's begin by understanding immunotherapies and immune resistance. So we know that immunotherapies have emerged as promising treatments for advanced cancer. However, immunotherapies such as immune checkpoint inhibitors are not always efficacious, and a subset of patients exhibits resistance to such therapies. One of the key barriers that limits this therapeutic efficacy is the immunosuppressive mechanism, which involve a diverse range of these cell types and their interactions within the tumor microenvironment. Additionally, hypoxic tumor, which is a common feature in solid tumors, and it's characterized by regions of tumor with insufficient oxygen supply, may also induce immune suppression as well as modify the metabolic landscape. So together, the spatial landscape of tumor infiltrating immune cells, tumor hypoxia, among other molecular and metabolic signatures, plays a critical role in treatment response. Therefore, characterizing this overall tumor immune landscape, along with understanding the associated immunosuppression, are critical for developing effective cancer treatments. And it's important to remember that the tumor microenvironment contains numerous distinct immune cell types that can either support anti-tumor immune response, protecting us against tumor cells, or they can have opposing functions and behave as pro-tumoral cells, thus promoting immunosuppression and tumor progression. And there's not one, but a large variety of these immunosuppressive cell types, such as MDSCs, TAMs, Tregs, that you can see on the screen. And these do not exist in isolation, but in a very complex interplay with cell types associated with anti-tumor immunity, such as tumor infiltrating T cells, of different kinds, dendritic cells, NK cells, to name a few. And especially when it comes to thinking about the main aim of cancer immunotherapies, it should be noted that there's this fine balance between this pro and anti-tumor immunity that's shown in red and blue here, which is predictive of cancer prognosis and the overall response to therapy. And therefore, characterizing such a complex landscape requires a multiplex approach where we can simultaneously visualize and quantify a majority of such cell types. So to interrogate the intricacies of the tumor immune landscape, we choose CellDive multiplexed imaging solution. Briefly, CellDive enables high-plex imaging within whole tissue sections at a single cell level. And using an iterative staining workflow as shown here is the backbone of the CellDive process. Specifically, it can map about 60 plus biomarkers from just one tissue section. And it's proven to be very gentle. So at the end of your studies, your tissue is still intact and can be used for downstream processing or for more experiments in the future. Finally, our patented two-step antigen retrieval has been shown to provide better antibody staining and as a result, maximizing the staining efficiency. Now let's get an overview of the CellDive workflow. We begin the process with our tissue sample. The first thing to do is to prepare these slides using a well-defined preparation process that includes a patented antigen retrieval technique, as well as more standard processes like devaxing, clearing, etc. And after the slides are prepared, we put them on the imager and use very quick low mag scans, so low magnification scans, to define the region of interest that will be imaged for the study. And when that's complete, we can get to the iterative part of the study, which is the imaging rounds. For this, first we stain with up to 4 dye conjugated antibodies, and we perform our biomarker imaging, and then we inactivate the dyes or turn them off with our chemical dye inactivation solution. And this process doesn't really remove the antibodies or photo bleach the sample, so it's very gentle and tissue preserving. And this is the step that allows us to stain over and over with new antibodies. And when that's complete, we image our autofluorescence signal again, and these are used for the computational autofluorescence removal. And this all concludes one imaging round. Those 4 steps are then repeated for different biomarkers until the study is complete. And from there, the images can be used as input for further image analysis using AVIA software to do comprehensive segmentation, classification, and spatial analysis to answer some of the key research questions. And here's what the iterative staining and imaging through a few rounds looks like. This is showcasing our collaborative work with cell signaling technology. And this is a CST panel of immuno-oncology biomarkers. To begin, we can see some beautiful signal from CD3 positive T cells, immune checkpoint markers like CTLA4, PDL1, and PD1 within round 1, followed by CD31 positive endothelial cells, CD11b positive myeloid cells, CD206 positive macrophages, and Ki67 positive proliferating cells within round 2. And this is followed by tumor immune markers like CD47, mesenchymal marker vimentin, dendritic cell marker CD11c, and hypoxia marker GLUT1 in round 3. And we can continue staining and imaging for more rounds to determine various cell types within the whole tissue. And after approximately 7 rounds, we generated some really high quality, large imaging data sets from whole tissue sections using CellDive. Here we see multiplexed whole tissue image that was generated from FFPE section of a syngeneic mouse tumor model of colon carcinoma origin. This tissue section was iteratively stained with about 30 CST antibodies to detect key proteins in the TME, or the tumor microenvironment, enabling immune cell detection and for further analysis. If we look at a zoomed in section of the tissue, you can realize how multiplexed whole tissue imaging can be utilized to probe key molecular players within the tumor microenvironment. Here we can clearly see the heterogeneous expression patterns of various immune, tumor, and metabolic markers, which we'll explore a little bit later. And if we keep zooming in, we can really appreciate how CellDive enables hyperplex imaging and exploration at a single cell level. This multiplex study can be extended to other tumor tissues originating from various sources. Here, I'm showing a few more data sets from syngenic murine models associated with various cancer types, like colon carcinoma, lymphoma, and breast carcinoma. Now, using this large panel of immune markers and their co-expression patterns, we can start looking at phenotypes such as cytotoxic and helper T cells shown here. And we can also see how other lymphoid cells, such as B cells, in the same field of view appear. But when it comes to looking at the entire panel of biomarkers and uncovering hidden insights from this data set, this starts getting a little complicated. Also, we're now looking at the whole tumor tissue overall, which may have another level of heterogeneity in terms of the spatial patterns or localized expression of some of these markers in distinct regions of the tissue. Specifically, this makes us think about some of the following key questions when it comes to gathering insights from such data sets. Number one, what are the molecular signatures associated with the tumor microenvironment? Two, what is the relationship between the metabolic markers, immune subtypes, and tumor cell types? Three, what are the spatial interactions associated with the immunosuppressive cell types? And to answer these questions, we'll find out how multiplexed image analysis powered by AI may allow us to map the tumor immune landscape. And to answer some of these questions and begin this process, we utilized AI-assisted analysis of multiplexed imaging from the colon carcinoma tissue using our AVIA software. The first step is segmentation. AVIA allows segmentation using the multiplex cell detection recipe, which uses all the specified channels that localize to the cell and the nuclear membrane to give individual cells and their nucleus as separate objects that can further be analyzed as shown here using the outline cells and the nuclei here, as you can see. The next step is to perform phenotyping, which deals with assigning biomarker positivity based on the antibody staining to individual cell membrane or nuclear membrane objects, which were created using the segmentation step. And here, for simplicity, shown in yellow and red are two cell types. Upon performing AI-powered phenotyping using a few cells for CD45 positive immune cells and CD31 positive endothelial cells for training, we get the following object, following output instantly. And the output of phenotyping is cell objects that are distributed into distinct classes based on measurements. For phenotypes like CD45 positive immune cells and CD31 positive endothelial cells shown here, it is fairly easy to classify them. But when we're looking at 25 plus biomarkers within the same study, especially where the same cells may co-express multiple markers based on the lineage it belongs to, it can get a little complicated. So we need some kind of clustering algorithm in order to characterize such cell types appropriately. Here we see that within this category of CD45 positive pan-immune cells in purple, we can see a few CD11b positive myeloid cells. And within this lineage, we can see these CD11b positive and CD206 co-positive M2-like macrophage populations. This brings us to step 3, which is clustering analysis. Clustering analysis allows quantification of cell types that co-express certain markers, and it may help us group cells by their biological function, which is extremely critical to understand some key players within the tumor immune microenvironment. Using this workflow on Avia, we then performed phenotyping for a set of cell types of interest. The clustering dendrogram from colon carcinoma tissue that's shown here reveals multiple immunosuppressive cell types within the whole tissue. More importantly, some key insights demonstrated in this clustering heat map show the co-expression of functional markers that may contribute to tumor immune resistance. Specifically, we note the presence of immunosuppressive regulatory T cell markers, along with expression of immune checkpoint markers like CTLA4, PD1, which may possibly be serving as an escape mechanism from the immune system. This is because this kind of immune cell population is typically associated with immune evasion and poor clinical outcomes in cancer patients. Next, we also note the co-expression of specific immune checkpoint proteins like PD-L1 and TIM3 in conventional dendritic cells, which are shown here in orange. And this may possibly impact their anti-tumor response. Further, we also looked at actively proliferating immune cells, shown in green here. And we find that these cells might have a large population of myeloid subsets of neutrophils or MDSCs based on Ly6G expression. Finally, in blue, we show that CD11b positive myeloid cells and specifically tumor-associated macrophages, or TAMs, show enrichment of GLUT1, which is a marker of tumor hypoxia. In summary, we find various tumor-associated immunosuppressive cell types. But this makes us think about the next question. What about the other markers and their interactions with the tumor microenvironment? And using unsupervised clustering on Avia, which uses phenograph-laden algorithm, it is possible to identify clusters of immune cell populations and their interactions within the whole tumor tissue. Here we can see how we can utilize all the markers and identify key interactions and immune cell clusters. One such immune cluster is highlighted in green. And here we see in green T cell clusters showcasing immune checkpoint protein expression of CTLA4, TIM3, PD1, along with markers of regulatory T cells, including FOXP3, IL2RA, which is indicative of potential immunosuppressive mechanisms impacting T cell function in the two tumors shown here. And these T cells are further showing interactions with other immune cell types, such as helper and cytotoxic T cells in cluster 9, and B cells in cluster 6 within the lymphoma tissue that's shown on the left side, and the dendritic cells co-expressing immune checkpoint markers in cluster 14 and 9 within the colon tissue shown on the right side. And this indicates some kind of heterogeneity within the tumor microenvironment, where distinct regions of the tumor exhibit differential immune cell infiltration and possibly different immune evasion mechanisms. Next, we can also observe another critical cluster associated with myeloid cell lineage, shown in pink here, which express M1 and M2-like functional markers for macrophages, such as INOS and CD206, respectively. At the same time, we also note PD-L1 co-expression in these cell types, which further suggests a myeloid-based immunosuppressive mechanism. And also, again, we note the enrichment of GLUT1 in these myeloid cell populations in both kinds of tissues, which is possibly aiding towards tumor immune evasion and resistance to therapy. Based on the insights from the clustering analysis, we identified spatially distinct heterogeneous expression patterns, especially for tumor hypoxia marker GLUT1 that's shown in cyan here. And it's important to remember that nutrient and oxygen availability are not homogeneous within the tumor microenvironment, and pockets of nutrient and oxygen depletion or hypoxia resulting from differential fuel uptake by heterogeneous tumor cell population is highly possible within the tumor. And this is evident from the expression pattern of GLUT1. Such conditions may result in immunosuppressive phenotypes, and they may limit therapeutic efficacy of a given treatment in cancer patients. What we see is, based on the insights from the clustering, as well as images that we see here comparing GLUT1 positive and negative regions, we note that GLUT1-rich regions seem to be enriched in specific cell types or immune cell types. For example, vimentin, shown in green here, CD11b, shown in pink, CD206, shown in yellow, CD47, shown in red, but not so much in Ki67 positive cells that are shown in white here. And this makes us wonder if there are specific immune cell types that are enriched within GLUT1-rich versus GLUT1-poor regions of the tumor tissue. Using k-means clustering on Avia software, we clustered cells based on the level of GLUT1 expression in each cell based on the mean intensity per cell. Here you can see the red, green, and blue colored cells that denote high, medium, and low GLUT1 expression. That's pretty evident from these images. And in the colon carcinoma tissue, this clustering by GLUT1 expression levels revealed differential enrichment of all CD45 positive immune cells. Interestingly, what we see is that the GLUT1 high regions or the GLUT1 rich regions are enriched in myeloid cell populations, such as CD11b, CD206, CD11c, along with specific immune checkpoint protein expression. Also, we can see the expression of markers associated with mesenchymal cells, such as vimentin, and immune evasion, such as CD47 expression, may indicate tumor adaptation and resistance developed within these GLUT1 rich regions. Also, it should be noted that this increased infiltration of myeloid cells, as well as immune effector molecules that we see here, such as iNOS and granzyme B, within the GLUT1 high regions, they suggest an active immune response within the GLUT1 positive regions. At the same time, we should also see that within the GLUT1 low or the poor regions, we note this enrichment of specific T cell markers such as CD3 positive, CD8 positive cells, and marker for regulatory T cell activation, IL2RA, along with, again, some immune checkpoint protein expression. Then there are also cells like B cells that are involved in anti-tumor immunity, and they're possibly found in both GLUT1 rich and GLUT1 poor regions. Therefore, using this analysis, we get a sense of what immune cell types might be associated with increased GLUT1 expression, but we still don't know if there's a major increase in the in the number of some of these major immune cell types within the GLUT1 positive regions. Therefore, we quantified the expression level of some of these major cell types identified within the colon carcinoma tissue. And we found that GLUT1 rich regions within the colon carcinoma tissue showed higher levels of immune cell subtypes, such as dendritic cells that are shown in blue here, M2-like macrophages shown in pink here, B cells that are shown in orange here, and a lower level of T cell populations compared to GLUT1 poor regions. So the T cells are shown in green here that seem to be decreased in the GLUT1 positive region. Specifically, M2-like macrophages are abundantly expressed in this tissue, and we can actually confirm their expression patterns and how it relates with GLUT1 localization within this tissue. And we can kind of appreciate that by looking at these images here. So GLUT1 is shown in cyan, and we can kind of see this overlaid expression of CD11B, which is a myeloid cell marker, and CD206 co-expression, which defines M2-like macrophages, or TAMs, also called tumor-associated macrophages. Finally, since TAMs or M2-like macrophages were among the major immune cell population expressed in the tissue, we also performed this spatial relational analysis on AVIA, where we estimated the distance between all cellular objects that are GLUT1 positive to all objects in macrophage category. And spatially, here we see a heat map of those distance relations, and it clearly shows the close proximity of TAMs to GLUT1 regions, almost creating kind of hubs of macrophage populations that are localized in these GLUT1 high or GLUT1 rich regions. It's important to note that such immunosuppressive phenotypes, like tumor-associated macrophages and their increased expression in these GLUT1 rich areas, can be associated with poor prognosis, and this may limit the therapeutic efficacy of a given treatment in cancer patients. So it's extremely essential to identify where they're located spatially within the tissue. And in summary, what we find is that cell-dive multiplexed imaging allowed this larger study of the tumor immune landscape and the molecular characteristics, specifically as delineated by the expression of the hypoxia marker GLUT1. And we know that GLUT1 is intricately related with the metabolic reprogramming, and the regulation of the immune activation within the TME. Also, using clustering analysis powered by AVIA, we demonstrated this differential enrichment of immunosuppressive cell types within the tumor tissues that we looked at. And using the spatial analysis, we also note the enrichment of tumor-associated macrophages, or TAMs, within GLUT1-rich regions, and T-cell populations such as Tregs within the GLUT1 poor regions. Localizations of such tumor-associated immunosuppressive cell types are, again, critical to identify, as they may contribute to therapeutic resistance by promoting a pro-tumorigenic environment. Overall, these findings could provide mechanistic insights into the tumor immune microenvironment and really guide treatment approaches by further interrogating the functional role of such immune cell subsets within the GLUT1 positive and GLUT1 negative regions. And understanding the association between tumors, immune cells, and metabolic reprogramming within the tumor microenvironment. And this might require further probing of the tumor tissues of different origins. And we looked at a few in this study, but this could be extended to even more tumors from different tissue types. And investigating the combination therapies targeting both glucose metabolism along with immune pathways. And finally, I want to conclude by saying that the spatial approaches mentioned in this study may significantly help in the development of therapeutic combinations targeting such immunosuppressive pathways. Before I end, I would like to sincerely thank our team at Leica, particularly our lead scientist, Arindam Bose, and Sophie Strubble, who work at Leica, a Boston Innovation Hub, as well as some of the key members of our cell dive team and the AVIA team who are mentioned here. I would also like to thank our collaborators at Cell Signaling Technology, or CST, for their shared effort on this project. And with that, I would like to thank everyone who joined the talk today, and I can take any questions.
All right. Well, thank you so much, Vasundhara, for your informative presentation. I would also like to welcome Arindam Bose, lead scientist at Spatial Proteomics at Leica Microsystems, who's joining us here to help answer your questions. So with that, we will now start the live Q&A portion of the webinar. And to our audience, if you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. Our first question here asks, what kind of antibodies can be used with cell dive?
I would like to answer that question, Kelly. Thanks for the presentation, Vasundhara. So as Vasundhara mentioned, cell dive can image 4 channels at a time, along with DAPI. And those channels are FITC, Psi3, Psi5, and Psi7. To increase the throughput, we prefer to use direct conjugates. However, we can also use primary, secondary to stain our tissue and image in cell dive.
Great, thank you. All right, next question here. Is it possible to reprobe the same tissue and add more antibodies to extend the study?
Yes, of course. So we have a patented dye inactivation protocol, which we can use to completely wipe off the biomarker dyes on one round and then label the tissue with second round of biomarkers, and by which we can do the multiplexing. In cell dive, we can do we can image one single tissue slide with more than 60 biomarkers. So yes, multiplexing is completely possible, and we can reprobe the same tissue with more antibodies.
Great, thank you. We've got some great questions coming in here. This next question asks, what is input for segmentation and clustering on AVIA?
I can take that question.
Vasundhara, we're not able to hear you anymore.
Are you able to hear me now?
We are, but it's very faint.
How about now? I'm just getting.
It's a little better. Yeah, can you just speak a little closer? Thank you.
Yeah. So for segmentation, the goal is to identify individual cell types and create those cell and nuclear membrane objects. And in order to do that, on AVIA, we use the multiplex cell detection recipe, which uses the CellPose algorithm. So CellPose is a generalist deep learning algorithm, which utilizes one nuclear membrane channel and multiple cell membrane channels. So it's particularly useful for any kind of spatial analysis where you have cell types that are expressing different cell membrane marker, and you want to essentially segment all the cells that are within your tissue. So as an input, again, for the CellPose and the multiplex cell detection recipe, which does the segmentation, it uses your DAPI channel as the nuclear membrane input and all the cell membrane channels that you have within your study. And you can also specify certain parameters like the sensitivity of thresholding for the nucleus and the membrane. And you can also provide inputs for the size of the nucleus, so the minimum size of the nucleus in terms of the area, as well as the average diameter of the nucleus. And then the second one would be what is the input for the clustering on AVIA? So clustering on AVIA is possible in two ways when we're talking about automated clustering. And one of them is k-means clustering, which is a pretty well-known method that is able to classify different objects based on a specific number of clusters that the user provides. So this is particularly helpful if you know how many clusters that you're looking for within your data. But a lot of times when you have a study that has 50 plus biomarkers, a lot of times users don't know how many cell populations or clusters we're looking for. And in that case, phenograph laden clustering is also available on AVIA, which can identify clusters based on measurements. So the measurements could be either intensity of the cells, which is the staining signal from the cell dive microscopy, and then there is also other parameters that can be used, such as spatial distance that can be used to cluster the cells. And depending on whatever measurement that's specified, you can perform this clustering using either k-means or phenograph laden clustering. So yeah, that's like a longer answer, but I hope that answered that question.
Great, thank you. Yes, we appreciate the detail there. And speaking of clustering, if we're segueing on that, we have another question here. How does auto clustering benefit the user for multiplex analysis?
Great. I think that's another great question. So again, I think what I'm trying to stress here is that a lot of times the users may not know before the study that, okay, I'm specifically looking for, let's say, T cells or CD3 positive T cells. So that's one way where you can do AI-guided phenotyping, and you specify those specific markers that you're looking for, and that's possible, yes. But when we want to find hidden insights from our data set where we don't know that there could be like multiple cell types that the user might miss out on if they don't specify it. So in that case, auto clustering is essential because it basically takes all your measurements, so all the cell types that are present in your sample, and then clusters them by their biological function, co-expression, spatial distance, depending on what the user specifies.
Great, thank you.
Mhm.
Right. Next question I have here asking for tissue preparation for cell dive, cryo or FFPE.
So CellDive is completely compatible with FFP tissue slides. We have also validated fresh frozen on CellDive. So we can use like different type of cell dives in CellDive.
Great, thank you. All right. Next question, how long does it take to stain and do the imaging for a study using 50 plus markers? Also for one cycle of staining, washing, and imaging, how long does that take?
Oh, that's a good question. So basically, in CellDive, in one round of biomarker study, we stain 4 markers, right? So again, the biomarker labeling time is not that long. Like it takes 1 hour to label the tissue, and all our antibodies are direct conjugates, and the dye inactivation is also very. It takes like 15 to 20 minutes to dye inactivate it. The main time is takes to stain to image the tissue, and that depends on how big the tissue is. However, if you have a tissue section of say 1.5 centimeter times 3.5 centimeter, it usually takes like kind of close to 1.5 to 2 hours. But again, you can select specific FOVs inside a big tissue section. Another advantage of CellDive is associated with a robot which is called bioassembly bot. So you can actually process multiple slides at a time. So you can basically load all your slides inside the robot. You can program the slide. You can load all your antibodies, all the reagents like PBS, mounting media, dye activation solution, and then you can start the robot and you can just leave the lab, and next morning you come and you see everything is done. So that's a big advantage of CellDive, which can process 15 slides at a time with the help of a robot.
Great, thank you. All right. Next question here asks, how do we test incorporations of new markers or biologic markers, antibodies into CellDive?
Yeah, that's a great question again. So we are partnering with cell signaling technology, and we have validated. We have a list of more than 350 validated antibodies, which works in CellDive. So we usually share that list with our collaborators, our customers. However, if you want to validate a new biomarker, we can definitely work together, and we help you to validate a new biomarker on CellDive.
Great, thank you. All right. Now, again, I want to thank our audience for all these great questions coming in. We do have some more time, so we'll continue with a few more questions. This next question asks, the cell-cell distances calculated is edge to edge or center to center?
I can take that question. So there are different ways to calculate that. It can either be through centroid to centroid, it can be through vertex to vertex, and both are possible on AVIA. I hope that answers the question.
Great, thank you. All right, another question here. Data from CellDive is in 2D. How well the AVIA work with 3D volume data?
That's another great question. We have looked at neurons in 3D, which are also non-conventional looking cells, and AVL works really well even with those cell types in 3D, which is, I think, a big advantage when you're looking at such irregularly shaped cell types and the way it performs in segmenting those in 3D. And you can also do more of these spatial analysis in those 3D samples as well, like what I discussed for any of this 2D data set that we went over today.
Great, thank you. And I see one question here asking if this video is going to be available to share with your colleagues. Yes, absolutely it will. You can share the same link that you used to register here. We highly encourage you to do so. All right, well, that will conclude our session for today. So I want to thank you again, Vasundhara and Arindam, for your time today. Do you have any final comments for our audience?
I would just like to say, thank you everyone who took the time out to, watch this webinar. And I would also like to thank the hosts here for organizing this talk. And I think the questions were wonderful. And thank you.
Yep. Thanks for joining. Thanks, everybody.
Great. Well, thank you both again for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Now, before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.